physical doctors. So today we are going to begin with our very first lecture on the epithelial tissue, which is a part of general anatomy. So when we begin up with epithelial tissue, basically, so what are the things that we need to know under epithelial tissue? Now we'll come slowly. So first of all, what are the characteristics of epithelial tissue? The constituent cells are closely packed. Closely packed together, and intercellular substance is reduced to minimum. Then the cells rest on a basement membrane. I hope you can understand. BM means basement membrane, made of amorphous substance. Then epithelial tissue contains no capillaries, and nutrition is derived by diffusion from the nearest capillaries. Then the surface cells are destroyed by wear and tear and are replaced by new cells. Okay, so these are the main characteristics of epithelial tissue. Okay, this may come as a question as a part of short note or anything like that. Okay, now moving on to the functions of epithelial tissue. Let's just change the color. Yes, so when we look at the functions, protection, from external environment, so protection from external environment. Second is absorption. Epithelial lining of alimentary tract leads to absorption of digested food, secretion. We know there are goblet cells in the alimentary canal. And respiratory tracts secrete mucus, provides protection, and minimizes friction. Fourth is lubrication, mainly in serous cavities. Example, peritoneal cavity, pleural or pericardial. Now, what we need to know are the types and examples. Yes. Mainly, it is of two types. See, you know all this from class 12 knowledge also, 11, 12 knowledge. But still, since this may come as maybe a short note, yeah, it's the most it can come is a short note. Still, I'm saying that the theory on glands is present. Okay, like types of glands and all that, it may not necessarily come. Okay, and whatever will come for exams, I will just cover it. Okay, whatever will come for exams, I'll cover it. So you don't need to worry about that. In compound, we have stratified and transitional. Stratified columnar, keratinized, non-keratinized. Now I'm not going much into the details of this. We will stick to some examples. Etc. Cuboidal, mainly glands and ducts of glands. Columnar, columnar is GIT, gastrointestinal tract, right? Male urethra, vas deferens. Then ciliated, mainly respiratory tract, right? Then pseudostratified, Pseudostratified is mainly mucous membranes of nasal cavity. Yeah, that's a nice example. Then moving on to transitional epithelium, we all know that transitional epithelium is mainly also known as urothelium because it's distributed in urinary bladder, then ureters. Okay, we went to stratified epithelium. Stratified squamous epithelium keratinized our skin. Non-keratinized mainly the mucous membranes of our body. Better would be to write that a tongue, mouth, cornea, etc. Stratified columnar mainly in some parts of mid-urethra. Now we go over to some more details. So first of all, we look at the now we look at the details of mainly the transitional epithelium first. So now we are going to discuss about the transitional epithelium. First, let us import a photo. Surface cells are large and rounded with abundant nuclei. Abundant cytoplasm and prominent nuclei. Surface cells are connected by tight junctions. I hope you know what are tight junctions, right? You have different types of cell junctions, tight junctions, desmosomes, etc. I hope you have studied it in class 11 and 12. This epithelium is best suited to line hollow organs which are variable in degree of distension. What does that mean? Like suppose an organ is present which may get distended later. Best example, the urinary bladder. It is normally it's not filled, it's quite compressed. When it gets filled, so it becomes distended. It increases in size. It blows up like a balloon, right? Mucous membrane of urinary bladder and ureters are made of transitional epithelium. It is also known as urothelium because of this reason. Allows rapid expansion of the viscous without stretching of the cells. Now moving on to some features. Possesses 3 layers: superficial, contains large polyhedral cells, then intermediate. As you can see, the upside down pear shaped cells are present. These types of cells. And the deep layer containing smaller cells. If you see the last layer, you can see very small cells are present. Now the surface cells perform 2 functions. Withstand stretching of distended urinary bladder okay. So when the cells are overstretched, a rupture of cells is prevented by cytoplasmic microfilaments and it possesses thickened osmophilic lamella which accounts for fluid impermeability. So the urine doesn't leak out of the bladder, right? The bladder wall is impermeable to fluids. So it is because of these features of the transitional epithelium. A short note may come from transitional epithelium. This information, if you can present it nicely, will be enough. So moving on to stratified epithelium. Mainly, we can see that stratified squamous epithelium is present. What are the features? Deepest layer is columnar. I hope you know this information previously. Succeeding layers contain polyhedral cells. Superficial layer flattened cells. This is a deeper layer cells are germinative. Now what happens that we can present a situation of two types of surfaces. On dry surface or I can write it here. It will be much better. On dry surface and on wet surface. On dry surface, the surface cells die and they get keratinized. On wet surface, the surface cells are alive. You can see they have a nucleus, nucleated surface cells. Okay. Now, what is the distribution? Epidermis of skin. What is the location? Tongue, mouth, lower pharynx, esophagus, anal canal, lower part, vagina, cornea, etc. So we are done with the stratified epithelium. Now we can have a look just at stratified columnar epithelium. Not much information is present. Superimposed fusiform cells found in some parts of male urethra. Not much information to it. Now we will discuss about the basement membrane. We all know that the cells of epithelial tissue or glands rest on a basement membrane. So it is the interval between epithelium and loose connective tissue. It is found within the connective tissue around muscle cells and capillary endothelium. It is composed of fine network of dense fibrillar materials. which lie in an amorphous matrix. A layer of irregularly disposed reticular fibers intervene between basal lamina or the basement membrane and adjacent connective tissue. So as you can see that it contains some parts which are mentioned over here in much details. I don't think we require, like lamina lucida, lamina densa, then the fibroreticular lamina, okay, which is mentioned over here. Right, all this is basically the basement membrane, these three layers. Some anchoring fibers may be present over here. Right, some collagen fibers, some elastic fibers. They're looking similar only. If we want to step into the details, though I don't think it is much required, but still, in case if a short note comes, you should try to mention this. It's good if you mention this and you can draw this diagram. So the reticular fibers beneath the membrane are argiophilic. Three layers intervene between epithelial membrane and connective tissue. A cell coat made of glycoprotein, a connective tissue proper, a basement membrane proper, and reticular fibers forming lamina propria. Basement membrane consists of three layers. So basement membrane consists of three layers. Right. What are they? The lamina lucida, Yes, it contains the lamina lucida, a lamina densa, and fibroreticular lamina. Basal cells are connected by hemidesmosomes to the lamina densa. And by anchoring proteins to lamina lucida. What are the functions of basement membrane? If I change the ink, supports the epithelial tissue, and imparts elasticity, some elasticity. Ionic or molecular transport of materials may take place across membrane. Right. Now we are going to learn about cell junctions. I'll use the same photo. There are also many cell adhesion molecules, also known as CAMs. They may be calcium dependent or calcium independent. So first of all, we are going to learn about the calcium independent ones. Calcium dependent ones. They are cadherins. So cadherins, strong intracellular adhesion. 5 heavily glycosylated external domains. Right. Ends are attached by linker proteins. To underlying cytoskeletal factors. Underlying cytoskeletal fibers. Now, mainly there may be different types of catenanes like E catenanes, N catenanes, P catenanes, right? But we don't need to go into too much detail. Now going on to selectins. They are found on leukocytes, platelets, and endothelial cells. Transmembrane lectin glycoproteins. Transmembrane lectin glycoproteins. Okay, so they mainly have carbohydrate groups. Okay, which mainly permit the movements. Like we know there are rolling and adhesion of leukocytes, WBCs, so those are all permitted by these carbohydrate groups. Then we go on to integrins. Glycoproteins, adhesion between cells and extracellular matrix, fibronectin, collagen, and laminin. Migration within matrix is facilitated by them. Integrins mainly are formed out of two subunits, alpha and beta. Right. Now we go on to the calcium independent adhesion molecules. That includes tight junctions, also known as zonula occludens. It's like a quintuple layered membrane continuous belt along the cell perimeter near apical surface. They mainly create diffusion barriers in continuous layers of cells like epithelia, mesothelia, and endothelia to prevent leakage of materials across cellular layer. Moving on to zonular adherence. Now, zonal adherence are not fused. A distance of around 20 nanometers is present between the two cells at this location. Form a belt-like zone of adhesion. Mainly cadherins are found in this zone. Now, macular adherence. Also known as desmosomes. We can see in the picture. See, desmosomes are present. They are separated. The adjacent cells are separated by 25 nanometer. They form anchorage points. Between cells subject to mechanical stress. Presenting actually not numerous in stratum spinosum of epidermis. Finally, we have the gap junctions. Also known as the communicating junctions. The gap present around 3 nanometers. Though you read more about gap junctions mainly in physiology, they are much more important from an aspect of physiology in your Devashish Pramanik book. Okay, much more information is given, but still, since it's a topic which is mentioned here, so I must cover it. Okay, mainly connections are present. Connections. These are the transmembrane channels which are present. 6 connexin proteins form this ring and contain a central pore. Passage of sodium, potassium and calcium ions and various second messengers. Except messenger RNA and other macromolecules. With that, we finish our discussion on the epithelial tissue.